So we're talking about cell reproduction. This is part 7. You only have a few more parts left, so try to stay focused. Do what you can to complete these courses. So we're actually going to skip around a little bit because there's only so much that we need to cover for this section. We're talking mostly about mitosis and meiosis with cell division. So let's see if I can get there. Present. We're moving to 10. So cell division happens through different phases. It is the taking of one cell, splitting into two offspring cells. So they're going to have the same DNA inside of them. It copies itself. It grows so that way it can form 2 new cells. So your eukaryotic cells have a specific type of division. So during this process, it divides both the nucleus and the cytoplasm. They divide. There are 2 types. There's mitosis and meiosis. Mitosis is for normal cells, so like your brain cells, your bone cells, your muscle cells, your blood cells. These are all done through mitosis. Meiosis is only for the sex cells. So cell cycle. In this series, it's the replicating events that creates life itself. Between cell division times is what we call interphase. So this is happening constantly. You're constantly dividing cells. You're constantly having new skin cells form on your body. Interphase is divided into 3 different phases. You have G1 phase, which is the growing of a cell. S phase, this is the phase where DNA is actually copied. And then you have G2 phase, which is where the cell is preparing for cell division. So G0 phase, the cell exits from the cell cycle and enters into this phase. During this phase, the cell does not copy DNA. It does not enter a cell division. So this is basically what happens at the end of cell division. And like it says, cells do not typically do this in nerve cells because nerve cells do not regenerate as well as the rest of your cells. So that's why if you bulge a disc in your back, or if you have nerve damage to a finger, you may not ever get that back. Mitosis. This is after interphase. Mitosis goes into prophase, which is the first phase. This is what starts. So if we actually go to here, we see prophase in the cell. This is your picture of prophase. So as this happens, the DNA shortens and coils. So it's shortening and coiling. That's why you can't really see it as well. Nucleosis and nuclear membrane disappear. So this is when it all that you can see. And then the centriosomes appear. Centrioles will be next to the centrosome. Both the centriole and the centrioles move to opposite sides of the cell. So as you see, it's starting to move. You can kind of see it. It's starting to move away. So there's your centrioles and there's your centriomes and centrioles. So they'll move to opposite sides of the cell. The spindle fibers develop to make microtubules. These come together to a mitotic spindle. So we're not going to worry about this. So keep on going. We're going to talk about metaphase. So metaphase is the second stage of mitosis. During this phase, the chromosomes move to the middle of the cell and line up. So if we move to metaphase, you see the chromosomes have moved, your spindle fibers and your centrioles have connected, and now they're getting ready to pull apart. Anaphase is the third phase. During this phase, the centriole splits. Each of the chromosomes move to an opposite side of the cell. So we split, we have the chromosomes moving to the opposite side of the cell. And in telophase, this is the last stage. After this, the opposite sides of the cell form, the spindle fibers disappear, the chromosomes return to a less coiled state, and now our nuclear membrane reappears and nucleosis reforms. And that's what you have here. Cytokinesis is when the cytoplasm will divide. And now we have our new cellular membrane forming. The same thing happens in plant cells, except it's much more rigid because of the cell wall. So you see prophase, everything's gathering. You see in metaphase, they're starting to pull apart. Anaphase, they pull apart. Telophase, you see the cell wall start to form. And then here's another way to look at it. See, interphase, everything's growing. Prophase, metaphase, everything lines up. Anaphase splits. Telophase, we have our two new set with cytokinesis. And of course, here is your cell cycle. So we go through G1, S phase, G2. Remember, G0 is only for nerve cells in animals. Then you have your mitosis, your prophase, anaphase, telophase, cytokinesis. This divides the entire cell, and then we move back into the cell growth stage. So you are constantly moving through all this. This is how cell division works. That's really all I want to cover. We can go through, so meiosis, it works basically the same thing. So during meiosis, cells go from being diploid to haploid. They still go through G1, S2, S, and G2. The difference between mitosis is that during meiosis, there are two divisions. So meiosis 1, they undergo the same stages. So prophase 1, DNA slightly coils into chromosomes. The difference is that homologous chromosomes pair up, and this is synapsis. Each pair is called tetrads, or tetrads. The chromosomes are lined up so that the genes of one chromosome are lined up with the genes of the other chromosome. During the synapsis of chromatids, a homologous pair of chromosomes twist around one another. Portions of the chromatids break off, join, switch to genetic material to the adjacent chromosome. So basically, this crossing over process results in the recombination of genetic material. So metaphase 2, the centrals line up. Anaphase, you still have, they move, split, moves to the opposite side of the cell. Then you have the separated at random. The results of random separation of mother and father genetic material produces genetic variation. So this is what we have when we combine our sperm and egg together to create something new. All right. And that's all today. So take the quiz. You're good to go. I hope all goes well. Remember, 7 out of 10. Move on to part 8.